lecture 2, part C on transmission genetics. This is the third and final part of this online lecture. So Mendel's experiments took place over many years, and they were much more complex than simply monohybrid crosses and test crosses, which we've just discussed. Once he had completed his researches on single traits, he focused on research involving two traits and how they could be transmitted simultaneously. So studying two traits required him to do a kind of cross called a dihybrid cross, di for two traits of interest. In the case of the traits that Mendel studied, although he didn't know it at the time, that meant two particular genes of interest, and it turned out that they were on different chromosomes. But he didn't know that at the time. So let's think about an example of a dihybrid cross. So here let's consider two traits. Let's consider seed color and also seed texture or shape. Okay, so the two possibilities for seed color are yellow, shown here, and green, shown here. Okay, and in this case, yellow is dominant. Okay, and it's associated, oddly enough, with the letter big G. Okay, and so in turn, lowercase g is recessive, and it's associated with the color green. The other trait that is being tracked here is seed texture. So pea seeds can be either wrinkled or they can be round, also known as smooth. Okay, wrinkled or smooth, wrinkled or round means the same thing. And in this case, it turns out that round is associated with the big W allele of a particular gene. Okay, and lowercase w, which is recessive, is wrinkled. Okay, so we're tracking two traits at the same time. So as usual, Mendel started with true breeding parental organisms. And the parental organisms that he started with are shown here. Over here, big G, big W, which is a plant that makes yellow seeds that are round, and little g, little w, which is a plant that makes green seeds that are wrinkled. So over here, he just happened to have an organism. I mean, it wasn't really accidental, he bred them this way, but he happened to have an organism that was parental, that was homozygous dominant for both genes of interest and both traits of interest, and he mated it against something that was homozygous recessive. Okay, so that's the cross on the left. On the right-hand side, he used an alternate configuration, but the same idea. He mated organisms that were true breeding for yellow and wrinkled seeds against organisms that were true breeding for green and round seeds. And these are the genotypes associated with those individuals. So, working from left to right again, this organism can only make gametes that contain big G and big W. Remember, these genes I said were on different chromosomes, so inside those gametes you're going to have a chromosome that contains the G gene, and it's going to be the big G allele, and another chromosome that contains the W gene, and it's going to be the big W allele. This gamete, which is all that this organism can produce, will contain the same two chromosomes. One will contain the allele lower G, or the G gene, and the other will contain the W gene, and it'll be the lowercase w allele. Okay, when these come together through fertilization, they make an F1 organism, first filial generation, and it is heterozygous for both of the genes of interest. And we see the dominant phenotype in regards to both traits. Yellow with regards to seed color, and smooth or round with regards to seed shape. And the same thing is happening over here. So when you mate true breeding parental plants that show the opposing phenotype for the two traits of interest, and you've got two traits of interest, we call that a dihybrid cross. All right, Mendel continued to use the same sorts of experiments. So he allowed the F1 generation, which are shown up here, he allowed them to self. Okay. And then he looked at what happened in their offspring, the F2, second filial generation. When the F1, which is heterozygous for both of the genes of interest, forms gametes, it can form 4 possible gametes: big G, big W, big G, little g, big W, little g, little w. Okay? And if it's selfing, then essentially it's mating against something with identical genotype, which could produce identical gametes. Each of those would be produced with a probability of 1/4. Okay? 1/4, 1/4, 1/4, and 1/4, and so on down the vertical axis. When you put these two together, you get that possible offspring. So there are all of the genotypes and phenotypes in that Punnett square associated with the F2 generation obtained from this selfing experiment, and their ratios are written out here. What you would see phenotypically is a 9:3:3:1, okay, relationship, and this is always going to be the homozygous recessive. This is always going to be a phenotype. This is always going to be the dominant phenotype, and then these will be a mixture, right, where half of them will be the dominant phenotype for seed color and the recessive phenotype for seed shape, and the other half will be the alternate configuration. So now let's take a minute and think what would happen if instead, as Mendel did do, he test crossed this organism. What would that mean? And by test cross, what do we mean? Let's look at that in the next slide. All right, let's run through this. So I just asked you, what would happen if you test crossed that F1? So here's the F1, big G, big W, little w. Okay, and we're going to test cross it. The tester is going to be homozygous recessive for the genes of interest. G, G, W, W. Okay, and by the way, its phenotype is going to be green and wrinkled, right? The recessive phenotypes. Okay, so let's make a Punnett square just to make this really simple. Okay, so let's put the gametes from the organism in question across the top, just because it's convenient for space in this figure. What gametes can this make? It can make 4 possible gametes, all possible combinations of G and W, 2 to the 2. There are 2 heterozygous gene pairs here. So it can make big G, big G, big G, big W, okay? Big G, little g, big W, and lowercase g, lowercase w. Can make each of those. Okay, those are the 4 possible gametes. I'm circling the gametes, okay, and each of them would be produced with a probability of 1/4, assuming the genes G and the genes W are on 2 different chromosomes, okay, due to independent assortment. What kind of gametes can the tester make? Well, there's only 1 allele of each, so all it can make is this, okay? So what do we get? It's a 1/4 Punnett square. It's really simple, okay? Big G, big W, little w, big G, little w, little g, big W, little w, right? Lowercase g, this was supposed to be underlined too, lowercase w. Okay, This is homozygous recessive. It's green. It's wrinkled. Okay. This is green. And it's smooth. This is yellow. And it's wrinkled. Okay. And this is yellow. And it's smooth. So what do we see? I count 4 different phenotypes there, and they're going to be present in a 1 to 1 to 1 to 1 ratio. Okay? And that is characteristic for test crosses against the F1 when you are studying a dihybrid cross system, okay, where you're tracking 2 genes of interest. There's clear dominance and recessiveness in the gene pair relationships you're studying, and each of those genes is on a different chromosome. So Mendel came up with his 4th postulate, which is the basis for Mendel's 2nd law, which we'll look at in a moment. And that 4th postulate was, during gamete formation, segregating pairs of unit factors assort independently of each other. This assumes the unit factors are indeed on different chromosomal pairs. This is because of the random nature at which sister chromatids align on the metaphase plate during meiosis. There's a meiotic underpinning to these observations. Okay, remember that handy formula that I gave you, which was 2 to the n, okay, where n is the number of heterozygous gene pairs that are being tracked in a particular cross. So if you're asking how many gametes can be produced, and you're looking at a dihybrid cross, n would be 2. If you're asking how many gametes can be produced, and you're looking at a trihybrid cross where you're tracking 3 genes of interest, then n would be 3. And 2 to the 3 in this case, or not in any case, 2 to the 3 equals 8. 8 possible gametes can be formed when you look at this organism, okay? So Mendel did trihybrid crosses and even, more complex crosses. And he would have started a trihybrid cross with the parental generation potentially like this, where all of the alleles, like all of the organisms, are homozygous. Now, I want to make a point here that you don't always have to have like all dominant and all recessive as shown here. It would have been perfectly appropriate to begin this cross as this one being on the left, being big A, little b, big C, big C. It's just that then you would have had to have mated it against essentially, its opposite, little a, big B, little c. Okay? You don't have to have like all dominant or all recessive for these hybrid crosses. So this organism here, this parent could only make this gamete. This parent here can only make this gamete. When the 2 fuse, you get this really neat heterozygote. There are 3 genes that we're tracking here, and we're assuming they're all on different chromosomes in these Mendelian genetic patterns that we're considering right now. How do you know how many gametes you get? You use the equation, okay, 2 to the n, and there are 3 gene pairs here. We did this in tutorial. And what are the gametes? Well, you have to look at all of the different combinations, and you have to have a systematic approach to develop it. They are all shown here in blue. And you have to go through and check them off and make sure that each are different and that they total to 8. This Punnett square would be an 8 by 8. It would be a 64 block Punnett square. So this starts to be a little bit harder to model when you're drawing your Punnett squares by hand. Okay, so hopefully by now you're pretty comfortable with thinking about these genes moving on chromosomes, and you understand the pattern of chromosomal movement during meiosis, and you understand that a Punnett square is a tool for visualizing crosses and their outcomes. Hopefully by now you also understand like what a gamete is and what is in there, okay? So having said all that, let me introduce another approach, okay? So there's an approach called a forked diagram that's really useful when we start to move from dihybrid crosses, which are really simple, into more complex scenarios like trihybrid crosses or even crosses where you're tracking more genes at one time. Okay, this method itself doesn't kind of mimic what happens during meiosis in quite as in a manner that is quite as easy to relate to the biology of meiosis, but it gives you the income and the outcome that you would get as the result of a cross. In order to work trihybrid crosses, it's really important that you understand the theory underlying monohybrid and dihybrid crosses. Okay, so you have to have understood and learned already that if you were tracking genes in a monohybrid cross, why I can say, and you should just understand, that the F2 will have a 3-to-1 dominant to recessive ratio when it comes to phenotype, okay, in a monohybrid cross. Because if you understand that, and you understand meiosis, then you can understand that when you're trying to track a trihybrid cross, you can figure out what's going to happen in the F2 of a trihybrid cross by breaking it apart and treating it as 3 separate monohybrid crosses that all come together at the end. Okay, so let's look a little bit about how that could be done. Here's an example of what the branch diagram or the fork diagram would look like for this cross. AA, BB, CC by the same. AA, BB, CC. Okay, each of these is lower case. Okay, picking up kind of from the previous slide. Okay, and we can break it apart into essentially 4 columns, each shown in blue here. The A, the B, the C, and then the combined column. And we're doing this, typically branch diagrams are used as a way of predicting what phenotype outcomes will be. It's a little more complicated and bigger and messier to do genotype outcomes this way. So again, we're breaking these apart when we use this method into 3 monohybrid crosses, an A cross, a B cross, and a C cross. We should just know as a fact, okay, have this memorized, that there's a 3-to-1 phenotype ratio in the F2 progeny of a monohybrid cross. So here we see 3/4, 1/4, 3/4, 1/4, 3/4, 1/4, and so on, so on, so on, and so on. There's your 3-to-1, okay? So here we've set up the first column and we're using the A's. 3/4 of them are big A, 1/4 of them is little a, okay? Then we draw a line from each of those and connect it to the B pattern. You need a lot of room to make these diagrams if you're drawing them by hand. Here's the B column. 3/4 are big B, 1/4 are little b, okay? And we're doing the same thing to show the possibility in this outcome here. 3/4 big B, 1/4 little b. Then we're handling the C, and this is where it starts to get really clunky, right? So 3/4 big C, 1/4 little c. 3/4 big C, 1/4 little c. 3/4 big C, 1/4 little c, and again, okay? And we line all of those up, and then we say, okay, what is the possibility of having an organism that shows the big A, big B, and big C associated genotypes, okay? And those associated genotypes can be calculated mathematically pretty easily using this forked line method, okay? So here we see this equation, 3/4, 3/4, and 3/4. This is what was associated with A, this is what was associated with B, and this is what was associated with C, okay? And this is maybe a little bit misleading, this figure, because this column applies to phenotype. And really the authors should have written the phenotypes out here instead of writing out these like 3-letter codes that look like genotypes for the gametes, because these are typically applied just to phenotypes. So what this is saying is that 27 out of 64, which is what you get when you multiply 3/4, 3/4, and 3/4, are going to have the big A phenotype, the big B phenotype, and the big C phenotype, okay? Similarly, 9/64, which is what you get when you multiply 3/4, which came from over here, 3/4, which came from over here, okay, and 1/4, which came from over here, so it's this path, right? That's what you get, 9/64. These will be organisms that have the big A and the big B associated phenotype, but the lowercase or recessive phenotype associated with C. If you don't believe me, write yourself a Punnett square, and it'll be an 8x8, and it will show 64 blocks, and see what you get. You can look at the Punnett square, and you will be able to count, and you will see that only one of the organisms modeled in that Punnett square is all recessive for the A, B, and C phenotype. Okay, 3/64 will be recessive for A and B phenotype and dominant for the C phenotype, whatever it is. Okay, so again, this is a bit sloppy here in that this is not showing us, not intended to show us the genotype of gametes, but it's rather to show us a simplification of the phenotype involved, make the slide cleaner. So here's some homework. I will be checking for this homework when we meet next. And I will expect to see it written out completely and neatly, okay, in an understandable way. The first part of the homework, which is shown on this side, is asking you to do the following test cross, okay? And the situation is written out fully here, and I expect that this answer will be pretty long. The second homework question is to consider this trihybrid cross, ask what gametes this organism can produce, how many of them and what kind, and draw out or write out their genotypes. That won't take you too long, I hope. And that is the end of this lecture.